Yep. Hi all. Is that loud? Is that better? I'm a new PhD student here at Uni Adelaide Animal and Vet Science School, and this is my project investigating the epidemiology of Toxoplasma gondii and macroscopic species of Sarcocystis in South Australia. And I'm here today to tell you why these parasites are important, but perhaps more importantly, what I hope to achieve over the next few years. Oops. Starting off with Toxoplasma, we know this parasite causes disease toxoplasmosis, and toxoplasmosis is important for wildlife, agriculture and humans. In Australian wildlife, toxoplasmosis causes neurological, respiratory and gastrointestinal problems, possible behavioural changes like we see down here with the cat and mouse, and mortality in captive and free-ranging populations. And within the literature, there's certainly the perception that marsupials are highly susceptible to this disease. In the field of agriculture, it's, the implications of toxoplasmosis are restricted to sheep and pigs, predominantly, where the disease causes fetus resorption, abortion, stillbirth and neonatal mortality. And in humans, toxoplasmosis can essentially cause everything I've just described and more: neurological, respiratory, gastrointestinal problems, possible behavioural changes, fetus resorption, abortion, stillbirth and mortality. And here we have a picture of a baby with hydrocephalus, or swelling of the brain due to toxoplasmosis. The implications of Macroscopic Sarcocystis is restricted to the field of agriculture, and within agriculture, sheep are predominantly affected. In sheep, Macroscopic Sarcocystis causes disease sarcocystosis, which results in carcass lesions like we can see in the... Whoops. I'm not used to these clickers. Carcass lesions like we can see in these two pictures. This is a diaphragm muscle of sheep and an esophagus, and the rice grain looking things are the carcass lesions. These lesions have to be trimmed during meat processing, or in the case of a highly infected carcass, the whole carcass is condemned for human consumption. This causes a reduction in carcass weight, a loss of quality product, a reduction in the potential market of the carcass, and additional labour costs borne solely by meat processors. But to manage and or mitigate these diseases, we need to know much more than just why they're important. We need to know about their biology and about their epidemiology. And both parasites are very similar in their biology. They're both coccidian parasites of felids, and they have three infective stages in their life cycle: the tachyzoite, the bradyzoite and the oocyst, each of which is pictured here, although the centre picture is actually a tissue cyst with many bradyzoites inside. So as I mentioned, the definitive host is felids, and in Australia we only have a single felid, the domestic cat. The intermediate host range is quite different for Toxoplasma and Macroscopic Sarcocystis. Toxoplasma is said to be able to infect all warm-blooded animals. This is mammals and birds. This is in contrast to Sarcocystis, that has a cat-sheep life cycle, the sheep being the intermediate host. If we look at their life cycle, they are both similar for Toxoplasma and Macroscopic Sarcocystis. This is actually a diagram of the Toxoplasma life cycle, but Macroscopic Sarcocystis is not too dissimilar. And they both start with the cat ingesting one of the three infective stages of the parasite, the tachyzoite, bradyzoite or the oocyst. In the cat, the parasite undergoes sexual reproduction and subsequently passes unsporulated oocysts in the faeces over here. These oocysts sporulate outside of the host and become infective, and when ingested by an intermediate host, consuming contaminated food, water or soil, they can cause infection and or disease. Inside the intermediate host, the parasite differentiates, forming bradyzooites and subsequently tissue cysts that last for the duration of the host's life. These tissue cysts can go one of two ways. Well, they stay in the intermediate host, but they can be consumed by a cat predating on wildlife, for example, and restart the parasite's life cycle, or they can also be consumed by a second intermediate host and cause infection and or disease in the second intermediate host. Both parasites can also be transmitted from mother to offspring via transplacental transmission for approximately 10 generations. So bringing this back to a more local scale now, if we look at both of these parasites on Kangaroo Island, we see similar patterns. And for Toxoplasma, we see that the seroprevalence in cats on Kangaroo Island is among the highest from any locality within Australia. Similarly for sheep, seroprevalence of Toxoplasma is higher than any other region in South Australia. And recently I've gone through SA Health's pregnancy and birthing records and found that for 2011 to 2013, the region labelled Fleurieu and Kangaroo Island had the lowest crude birth rate per 1,000 people, lower than any other South Australian region. This could be due to a whole number of factors, but I think this alone suggests that it's a pattern worthy of investigation. For those interested, I was unable to follow this pattern back any further because previous to 2011 the coding of the data on their website had changed, but I've asked for that to be recoded. This is Macroscopic Sarcocystis, and I've created this picture using PIRS's abattoir surveillance data, which is data collected on Macroscopic Sarcocystis at the Murray Bridge and Loberthal Thomas Foods abattoirs between 2007 and 2015. And as you can see in red, we've plotted all the sheep properties with a history of Macroscopic Sarcocystis infection. In blue, all the sheep properties with no history of Macroscopic Sarcocystis infection. And I'm sure you'll agree there appears to be quite a spatial cluster on Kangaroo Island. If we look at the prevalence of this parasite, we see again that Kangaroo Island stands out. And the graph on the left is all sheep lines that went through the Murray Bridge and Loberthal abattoirs between 2007 and 2015. On the right is all sheep lines with at least one sheep in the line with Macroscopic Sarcocystis infection, using the same data. And as I said, Kangaroo Island appears to be standing out. So why might this be? What are possible explanations? The first is high cat and or intermediate host densities on the island when compared to the mainland. High seroprevalence of these parasites and host species on the island when compared to the mainland. A greater availability and accessibility of carrion on the island when compared to the mainland, and or more favourable environmental conditions on the island when compared to the mainland. So this leads me into my PhD, which in a nutshell will attempt to answer these two questions. The first being, why are we seeing this pattern? Why is the prevalence of these parasites higher on the island when compared to the mainland? And the second, what is the economic impact of these parasites on the Kangaroo Island sheep industry? So to answer these questions, I'll compare a number of things between the mainland and the Kangaroo Island and the mainland. First being cat and intermediate host densities using camera traps, seroprevalence using a serological survey, and carrion usage using camera traps. Environmental conditions, haven't worked out how I'll compare it yet, but I'll work that out in due course. I'll use a maiden use survey to assess the economic impact of Toxoplasma, which will allow me to identify if ewes contracted the parasite during the pregnancy period, and again use PIRS's abattoir surveillance data to look at economic impact along with stochastic modelling. I'm running out of time, so I'll keep going. This project will have a couple of outcomes. The first being a better understanding of the factors that contribute to the higher prevalence of these parasites in host species on Kangaroo Island. This will be invaluable to disease management and or mitigation in wildlife, agriculture and humans on the island, but also aid in their management and or mitigation elsewhere. This project will also provide an accurate estimate of the economic impact of these parasites on Kangaroo Island, which will highlight the importance of managing these parasites. Overall, this project will have implications for wildlife groups and organisations, agriculturalists, human health organisations, land managers and landholders. If you want any more information, come talk to me in the break. Thanks.
Okay, any questions? Yes.
I'm not sure if it's a question or not, but it's just interesting when I was working on Kangaroo Island in 0506, they had a heavily subsidised initiative where they would microchip all pet dogs and cats on the island, and I think that was motivated so they could undertake tracking of feral cats. So it's interesting that the data that you had there was still showing such, potentially such high effects of cat.
Yeah. In terms of the Toxoplasma study showing a higher seroprevalence in cats on Kangaroo Island, that was done by Jack Redden, which was a few years ago now. I can't remember exactly, maybe 2005. And the seroprevalence in sheep was again a few years ago, but the Macroscopic Sarcocystis data, as I said, is from 2007 to 2015, so that's very recent. Both parasites are transmitted by cats, so I don't see why there would be not the same pattern if we looked at recent data for Toxoplasma.
Great.
Was the original data on prevalence from domestic cats or feral cats?
From memory, it was, well, traps Jack and other people, cats Jack and other people had trapped. So whether they were domestic or feral, they, yeah, I think so.
Okay, one more question. Okay, well, no further questions. Thank Patrick, and I would also like to thank all the other speakers for keeping so beautifully to time.